Great, thank you. It's a pleasure to be here, and thanks organizer to invite me. So we're trying to describe this new publication, which is a computational tool called Monopogen. We're trying to call both germline and somatic variants from single cell sequencing data, including DNA, RNA, and also attack-seq and potential other data type, which I will be, talking later. So my lab has mostly, at least one of the theme for us is to study cancer evolution. And I think increasingly people understanding this is a process that started very, very early on, actually since the normal or physiological normal tissue also can have somatic mutations. And over the time, the tissue can become precancerous and eventually become cancerous. And lots of mutations can accumulate in cells throughout this process, which is associated with the phenotypical transition of the tissue and definitely disease advance. So our key question has always been cell of origin. Basically, where does the cancer cells come from? What type of tissue or cell type are those cells initially derived from? And what are the clonal architecture? Are there multiple somatic clones or single clone dominating the population? And how did they get there? And what are the process? Maybe there's different pathways for the clone to evolve into the current status. And what are the drivers? Are they mutations or epigenetics or, epigenetics plus environment? So what are the particular important factors we can, that is determine the mechanism of evolution? And what are the context for those drivers to become drivers? And by understanding those, can we kind of intervene the developmental process and try to alter the course of the disease? So those are always our key scientific questions. And increasingly, people understand the somatic evolution process not only affect cancer, but also affecting many other diseases like Alzheimer's disease or cardiovascular disease. And so those are started to unfold as people have increasing access to the normal or precancerous tissue and be able to apply sequencing on those tissue to detect the somatic landscape of those tissues and also associate those with different type of disease and their characteristics. And for example, this study several years ago has shown not just clonal hematopoiesis, which are how the blood cells were derived from, but also in the somatic tissue like colon, liver, and other skin. And the mitral, they also have somatic mutations in their so-called physiologically normal tissue or cells. So this is a whole new space which is started to be explored by, for example, the SMART consortium. Okay, for us, we have started doing this very early on, about almost 20 years ago by now. So starting by doing the first cancer genome sequencing back in 2008 in one of the first AML genomes, where as the NGS technology were become popular at that time, we leveraged the high throughput to be able to actually sequence the whole cancer genomes and to detect mutations. And later on, we published a bunch of studies, using NGS at different level of sequence coverage and to be able to identify, for example, mutation or clonal evolution between two disease stage, MDS to ML transformation, for example, and over a course of time, and to be able to basically preliminarily observe the clonal architecture and their evolution over time. So a bunch of methods has been developed over those times. So were methods like MUTAC and others developed at Broad Institute and MIT and Harvard, for example. So those has been particularly powerful at the time, for example, in identify structural variants, different type of structural variants. And when you apply those into disease tissue, you can actually identify particular oncogenotypes like this. This is basically insertional translocation of this piece from chromosome 15 in 17 and creating a novel gene fusion PML alpha, which is kind of determines the APL diagnosis and also the therapeutics. So those are the early examples of how NGS sequencing is able to help perform personalized medicine. Okay. However, increasingly, we realize bulk sequencing is not the best approach. I mean, at least if you were to understand the cancer evolution at higher resolution, you have to use single-cell sequencing technology. That's why we collaborate with our collaborator, Nicholas Nevin, since almost 15 years ago to try to develop informatics or tools to analyze single-cell DNA sequencing data, from which you can identify a cell and mutation matrix with lots of missing value and also potential higher error rate. Nonetheless, those data is able to help us understand the clonal composition of a tissue at single cell resolution and able to construct final genetic traits based on the mutational profile, and also identify clones with similar mutational profiles. One of the tools we developed, as mentioned by August, is Monopogen, published almost 9 years ago, is to basically implement error model, much more or less a genotype likelihood model similar to the programs in the 1000 genome projects, and be able to compute a posterior probability to tell if a particular genotype has a high probability in a particular cell. So that's what this program generates is a VCF file with cell and mutations in each particular cell. So and many of you know that one of the foundation of our current genomics or population genomics is this 1000 genome project, which basically the last version, phase 3, was published in 2015. It has about 3,000 individuals sampled from across the globe, and 26 major ethnic populations and genetic populations. And it produced a pretty high quality haplotype for each of those human populations, which now can be used as a reference for us to trace the genetic origin of samples if the genotype data can be obtained one way or the other. So this is basically one of our reference population genetics database that is being used also in the Monopogen work as well. So nowadays, I think the reference idea has been expanded into the transcriptomics level. For example, the human cell atlas is now having a transcriptomic of different cell types, the characteristic transcriptomics of different cell types across 18 different organs, and which has been, as you guys know, has been tried to model by foundation, different type of foundation models. And those samples were actually taken from roughly also similar to 1000 genome from across the globe, and with hundreds, thousands of samples, actually a little bit less than the 1000 genome project. So one of the questions is, are the sampling sufficiently representing the diversity of a human genetic population, or they actually have particular type of bias in the sampling process? So those are actually very important questions to be asked. So that's why the Chen initiative, which is driving the Human Cell Atlas, has approached us, and they wanted us to develop a tool which can call SNPs and mutations from single-cell RNA attack sequencing data, which are the main transcriptomic reference data sources, and to be able to actually at least characterize the ancestral origin of those samples and be able to tell if there's a bias in the sampling process. So more importantly is to understand how the genetics makeups, for example, SNPs, are affecting the cellular traits. Do they affecting the gene expression level of different, people from different ethnic groups or race? As has been known, those are actually, has been shown, particularly in African American individuals who are predisposed to triple-negative breast cancer. I think those are clear cases of where SNPs could affect in the cellular traits. And also they actually help us remember those data are actually sequencing data, not just count data, which I guess most of the community has this kind of a understanding of this. Those are just count data focused on a small, tiny portion of the genome. But it turns out it's actually much broader than people has anticipated, which I'll show you later on. Okay, so with that being the background, we started to develop this tool called Monopogen, which intended to be able to call SNPs and somatic mutations from this kind of a pan single cell sequencing chemistry or techniques, including not just RNA attack, but also DNA, and also increasingly other data types. The tool has two different modules. First is germline modules. So this is largely solved by the imputation, the germline imputation, tools which are existing. For example, like the Beagle and other tools, much similar to the 1000 genome, low pass sequence and context, where you have to deal with low coverage sequencing of the reads in each individual cell. But because those cells are actually coming from the same sample with same expected haplotype, so those information can be actually borrowed from a population level from the 1000 Genome Project if we can figure out the haplotype that is within each individual cell. And we can use those information to basically impute the genotype likelihood to identify the most likely genotype at any given location. So this is a relatively easy in terms of a germline calculation, but it's actually pretty important, as I'll show you later. A more challenging subject is to identify the somatic mutations which does not exist in the reference, and they potentially being plagued by different type of error, both sequencing and DNA amplification error, as well as potentially different level of contamination or different type of clones being mixed together. So the statistical level of algorithm or approach we're using here is trying to examine relatively high coverage region of the data and trying to reduce the amount of errors through different machine learning modules to remove the low quality locations, and trying to leverage linkage disequilibrium LD structure in the data, where if there's a somatic mutation, we will be able to tell they are only affecting one subset of the cells and they consistently exist in one of the allele. So we use those statistics to be able to help us compute somatic scores, which help us eventually distinguish germline from somatic variants. So those are parts probably deserve more detailed discussion if you would like to, and we're happy to follow up with you to do that. Okay. So with those germline and somatic SV detected, you can do a lot of things. Basically, you can determine the genetic ancestry of the cell. You can also trace somatic clonal architecture and evolution. You can even potentially understand the function of those variants in the context of the cellular traits or cellular phenotypes. So actually, if you have any question, I have about 35 slides. I think you could even interrupt me, I think. Feel free to interrupt me. I'm happy to answer any question during my presentation and also at the end. Okay, so I'll just keep moving. So this basically illustrates the germline imputation process in a little more graphic detail. So if you have a location with very low coverage, one reads cover the location. However, you happen to have another location where you have higher coverage in the same haplotype as determined by 1000 genome reference database, then you can basically borrow information from this haplotype database to actually infer the alternative alleles that is likely exist in this cell. And this is how germline imputation was performed. And again, we did not develop anything new. We just used Beagle as a tool to compute this imputation. Okay, so So one part which has been very pleasantly surprising is the amount of coverage in those single cell data, sequencing coverage, which we also underestimated before we started this project. For example, we initially started with 4 sample of single cell, a single nuclear RNA-seq data developed by the Chen lab from the human retina cells. And as you can see, the amount of coverage or number of reads per cell is actually typical, relatively higher, 20,000 reads. And those are 3' 10x RNA sequencing data. So if you look at this data, you will see actually 40% of genome are covered by at least 1 reads. And 20% of the genome are actually covered by at least 8 reads. So it's much more than just 3' coverage is actually much broader than we thought it. So that's actually very good for mutation or SNP calling. So we perform what I described before in terms of germline calling on those data sets.
Professor Chen, I have a question. So
yes.
Did you see overall, you always see this kind of coverage, genomic coverage across different single cell RNA-seq data, or it's just for that single nuclear RNA-seq?
Yeah. I have a summary in the later slides. That's a great question. It's a lot varied by different data sets.
Yeah.
Sorry, that. Yeah, you're absolutely correct. So this one is actually relatively well covered single nuclear RNA-seq data, which make it relatively easy to begin with. As you can see, just by doing the big imputation based on 1,000 genome, we're able to very confidently detecting 20% of the whole genome SNPs. Which present in the single nuclear RNA-seq data. So this is actually pretty, encouraging at the beginning of the phase. And as you can see, what very interesting is a lot of SNPs detected are actually in the intronic region. Even though the sequencing were, it's not that surprising because this is single nuclear RNA data, which has a lot of intronic coverage as you can, imagine. But its number is still very much striking than other SNP detected from other regions. And the amount of coverage actually is pretty high for. Of course, this is cumulative across all the cells. It's very high actually in 3' UTR as expected, but also reasonable for the other type of genomic regions. And so this gives us a lot of SNPs to mapping the samples back to their ancestral locations through the principal component analysis using the HGDP SNP data sets. This is a population diversity panel data sets as a reference. As you can see, we can confidently map 3 of the samples back to the European origin and 1 of the sample back to the Hispanic origin. This is self-reported Hispanic as well. So basically this makes complete sense. And if you look at the chromosome punting results, which report specific chromosomal fragments in terms of their origin, colored by the origins, you can see the single nuclear RNA-seq has determined origin. The coloring pattern is very much consistent with those determined from the paired whole genome sequencing data. So this is basically validate the accuracy of the genetic mapping from single nuclear RNA-seq. And if you look at even this Hispanic with admixed ancestry, you can see the admixturing between green and purple are also very much aligned with what you saw from the whole genome sequencing data. Of course, you have some additional places of discrepancy, but overall, this works pretty well at even chromosomal fragments level. Okay, so we move on a more challenging data. This is a single cell RNA sequencing data, which is mostly focused on the transcript RNA in the exonic region. However, as you can see, again, a lot of the discovery or make SNPs were called from intronic region as well. So basically, even for single cell RNA-seq, there's a lot of coverage in the intronic region as well. And one of the interesting point of this is this study is focused on Asian population, Japanese, Korean, which has very much similar genomes. So a key question is, are we able to actually distinguish Japanese and Korean, which are evolutionary very close, as you know, from just single cell sequencing RNA sequencing data. So we were able to again confidently map those Japanese Korean back to the East Asia population. And also, interestingly, by unsupervised clustering, we're able to actually distinguish Korean from Japanese. So this tells us the density of the SNPs and also the coverage of the breeds of the SNPs are able to help us actually delineate, very close population. So which is actually very exciting as well. So okay, here's the summary. I guess August asked me that question in terms of different technology, in terms of a different number of cells in each data sets, and in terms of sequencing coverage, how different data sets differ in terms of SNP calling results. As you can see, different technology, nuclear RNA-seq, this was our example. Here we're actually compare against other tools available at the time. As you can see, for RNA ATAC-seq, we're doing very much clearly better than other tools based on the ROC characterization. DNA, we're actually similar to a few other tools. In general, when you have more cells, the detection sensitivity increases. But the genotype accuracy is relatively steady across, number of cells. So this is actually very much thanks to the imputation architecture, which has been implemented over the years by the 1000 genome. And single-nuclear RNA sequencing actually has a variety of different level of coverage. So as you can see, this really determine the detection rate or calling rate, a number of SNVs. But the accuracy is actually relatively has always been high. So it's so whatever being called, you can trust the accuracy of the data. So of course, ATAC-seq, even though it has relatively low coverage per locus, but it has broader coverage across the genome. So in total, you're able to get a lot of SNPs from ATAC-seq data than from RNA sequencing data. So this is actually kind of a summarize how technology and different level of parameters affecting the range of results you should be expecting from your data. Okay, and with that data, you can actually start to do different kind of a cell level, cell type level eQTL, QTL analysis. For example, here you can associate the whole genome SNP you imputed from the data associated with cardiomyocytes, transcriptomic traits. Here we actually computed the metabolic levels based on RNA expression data of cardiomyocytes. And then through the genome-wide association analysis, you identify those particular genes and locus which is associated with those particular traits. This has been reported before. So this is a validation of this, how useful you can apply this data into gene association analysis. Here's another example of calculation from ATAC-seq data. You can see you can calculate GATA4 transcription factor activity level based on the peak profile and sequence covered under the peaks. And then you can do the genome-wide association analysis and identify this particular gene, which is associated with this particular GATA4 transcription factor levels in cardiomyocytes. So those are some illustrative example of how this data can be broadly applied. Again, this is actually a 6.5 donor from ENCODE data, public available on the normal heart tissue. So you can definitely apply this to a much broader context in many of other studies. So we recently expanded this into tools with single cell or small indel detection. As you can see, this is the same data sets, and the indel, which has a panel at the bottom here, is able to similarly inform us about the ancestral origin of this set of samples consistent with SNPs on the left panel. So this tells us, now you can also do indel detection using similar framework and beyond just single nucleotide polymorphisms. Okay, and we apply this to the H10 datasets, which is a cancer single cell study. But also people are interested in how genetic ancestry determines or predispose cancer development. And those samples also had whole exon sequencing. And we found it's interesting to compare SNPs we can call from single cell sequencing versus whole genome bulk sequencing. As you can see, what you can call from bulk sequencing is actually a tiny fraction of what we can call from the single cell nuclei attack and RNA sequencing data. So we are able to call 20 or 30% more SNPs, mostly in the non-coding region, as you can anticipate. And those were very accurate compared with the whole genome sequencing in the coding region. And they cover 40% of the exonic region of the whole exon sequencing data. So therefore, this is actually a pretty useful resource. Okay, there's a question, please.
Yeah, it's my question. So Dr. Chen, just wondering, like, say here, there's more sleeps by using the single cell RNA-seq data. I was just wondering, like, what's the sequence depth for the whole exome sequence data? And then, I'm actually surprised, like, there's so many, sleeps we called by single cell RNA-seq data, not the whole exome sequence data. So if you can you actually tell us, the sequence depth for each, technology you have used here? Just wondering.
I don't have the specific number. Whole exon sequencing was done in a standard way. I would anticipate 40, 50x coverage, whole exon bulk sequencing. That would be the range. And single cell attack RNA sequencing, I think they are also relatively standardized. So maybe it's like what I showed in the previous slides. So I don't have a particular number, but as I explained to you, I think a lot of those were non-coding regions. And we benefit a lot from imputation against the 1,000 genome project. So even though the coverage is likely low in the non-coding region, but we can still borrow information from 1,000 genome to actually call those SNPs. So I guess that explains why you are calling much more SNPs than whole exon sequencing. So okay, so I'm gonna continue move on. So you can now actually confidently map those samples back to their genetic origin for those cancer samples. And as you can see, much of the samples are Caucasian or European ancestry, and a bunch of those are African Americans likely, but a few are actually East Asian and also Central Asian patients in the breast cancer. This is actually breast cancer cohort in the H10. And those mapping results can be pictorially described here. Purple are the European, and blue are the African ancestry patients. And interestingly, two patients self-reported as Asian, but our process actually delineate them as South Asian. So sometimes the patient record is incomplete, but genomically, based on the data, we're able to obtain further information about those patients. Okay, so I'm just switch gear into the somatics. Are there any questions regarding the germline mutation coding so far? Okay, if not, I'm switching gear to the somatic mutation coding. So as I briefly described to you previously, somatic mutation coding is much harder. We basically focused on the regions where there's a lot more coverage. I think we had a cutoff, I don't remember exactly, it's potentially 400 reads.
Yeah.
400 reads. If you have the whole data set has 400 reads cover a particular location, then we try to examine potential of having either a de novo germline not existing in the 1,000 genome or actually somatic mutation at those type of location. Of course, the 400 is distributed into different number of cells, which we will be able to resolve at a later stage. And we do different QC quality control. For example, remove cluster of SNVs, which typically representing low quality region or difficult region of genome. And we also remove based on a noise model we learned from the germline variant coding steps. And also more importantly, we introduce our so-called somatic or LD refinement score here, which help us to delineate somatic from germline mutations. The intuition here is that because if it's a true somatic mutation, it should occur consistently on the same allele, like the minor A allele, in different cells. If it's a random error, it probably occur in different alleles, not actually the same allele. And also, if the variant allelic frequency is lower, then it's more likely a somatic mutation than a germline mutation. So we're kind of combine those statistical characteristics of somatic mutation into our scoring system. And I just would like to point out, I think because of this calculation, our tool can actually delineate somatic mutation at this cell or allele level, right? We don't actually require any reference normal, cells or normal samples. We are able to automatically delineate those allele origins or assign the allelic origins to each individual cells. And we are able, and then further delineate if they are somatic or germline variants in a later stage. So this is actually a significant advantage, making our tool much more sensitive and also much more accurate than some other tools existing, which I'll show you why we think that's the case. Okay, we initially validated this using the Peter Van Ganen's data. This is a bone marrow sample having clonal hematopoiesis, differentiation of different blood cell types from the HSPC. And advantage of this data, it's multi-omic. It's single-cell multi-omics. It has transcriptomics, mitochondria transcriptome, and also TCR sequencing from the same cell, so which can help us validate our mutational coding. As you can see, we're coding roughly tens of thousands of de novo SNVs, and via different filtering strategy, we ended up with 300, roughly, hundreds of those putative SNVs. One way to validate this is to compare with mitochondrial clones. So if the cells can be assigned to different clones based on their mitochondrial mutational profile, and it can also be assigned to different clones based on the somatic mutations detected by our tool from RNA from the transcriptome. As you can see, those two types of assignment actually match pretty well. They basically assigning cells into similar clones, high concordance, 63%. And using this new RNA mutation, we're able to further delineate those clones which were not able to delineate by mitochondrial variants into different type of clonal origin, particularly in the erythroid lineage. Blue or actually now become blue and red. Some of the cells are potentially from different genomic origin rather than, even though they have similar cell type, but they actually have different genomic origin. Similarly, if we look at T cells, the TCR sequence are able to delineate subsets of T cells which has the same TCR or same T cell clone expanded during the differentiation process. However, if you look at the RNAs in those T cells, the mutations detected from the RNA of those T cells, you can actually see, additional origin of those cells. For example, this set of cells which potentially did not fail in the TCR sequencing, actually potentially have similar genomic origin as those cells, had the genomic sequencing. So therefore, this enriched us about the genomic origin of those cells, in this particular data sets. Okay, this is kind of a validation using single-cell multi-omic data to our mutational coding, somatic mutational coding. Okay, more recently, we started to have another experiments. Basically, this is data derived from SMART consortium. This is an in silico admixture of different cell lines. So this is 6 different human cell lines admixed at different level. So this Han Chinese wars are basically present at a very high level, 84% in the admixture culture. And those are basically mimicking subclones of tumors, presenting at 10%, and the real one is a 0.5%. So 6 cell line gets in vitro mixed together, and those were either sequenced, per individual cell line to derive the ground truth, or sequenced from this cocktail of in vitro mixture mimicking the tumor samples, and then performs somatic or germline and somatic mutation coding using Monopogen, and also another program called Schematic, published in the same year. The main difference between Monopogen and Schematic is in Schematic, the tool require you to to be able to separate cells into different bins. Those can be done by different cell type, by transcriptomics or by any other approach. So they require you to tell the program which cells are belong to which cell type or cell clusters in advance. And based on those separated cell cluster or cell groups, you can perform sort of a contrastive mutational detection using algorithm like MUTAC to identify the clone or cluster-specific variants. Using other cell clusters as a reference, as a background to identify those. So as you can see, it's a very different type of analysis. Ours are delineated at allele and single cell level, and theirs are actually performed at the cell type or cell cluster level. So I'll show you, this actually make huge difference. So from this data I just described, we caught mutations using Monopogen and Schematic. And in terms of a total number of SNVs called, we were able to call a lot more than Schematic. And a very high proportion of those are in the ground truth, which are individual sample whole genome sequencing data. So this is again indicating a pretty high specificity or accuracy of calling results. Schematic is considerably lower in this exercise. And if you look at this sample specific characterization of mutations, you can also see we actually detect a lot more sample-specific mutations are actually presenting in the ground truth. So the specificity is also, both sensitivity and specificity are higher than Schematic in this, benchmarking exercise. Okay, that's kind of further validating. And oh yes, please.
Hey, sorry, it's me again. I think one question. We actually also invited the developers of Somatic to present at BCH also, maybe a few months ago. So I'm glad that you actually compare your tool with their tool. That's great. One question I have here is, so for the Monopogen, so basically for what Somatic or germline calling, we basically call the variants for the whole cell population, not for the cell type specific group, right?
Mm-hmm.
Okay. Yeah, I think that's part of the reason why we have high accuracy. Looks like because somatic mutation, sometimes when we try to split the cells into cell type specific BAM files, the sequence depth is so low, and then for some specific cell type, if the cell population the number of cells is very low, it's impossible to call the somatic mutation at all.
That's a great point. And I just like to add to that is because we're doing mutation detection at the cell and allele level, so we do not assume the cell population segregation here is completely accurate. I think Schematic would require you to have relatively pure cell population in each BAM file. So therefore, when you contrast one against the other, you can have a strong signal to noise ratio. We don't have any of those assumptions. We basically don't split everything into BAM by empirical knowledge. We just automatically tell based on the linkage disequilibrium pattern in the data itself to tell which one is somatic, which one is germline. So this gives us a lot more power and both sensitivity and accuracy in determining cell type specific or sample, somatic subclonal specific mutations, as you can show here. So I just like to clarify on that point. But your point is also important. Sometimes you don't even have many cells in the individual band files. So that's also another potential limitation of using that type of approach. Okay.
Thank you.
Thank you for the point. So this is an independent validation, actually. Another study, as if you may be interested in this, we are not involved at all. So here, this is a 3 different type of sample health, a primary tumor, lymph node tumor. And they caught the mutation from, this is single cell RNA sequencing as well. And they construct the phylogenetic tree. As you can see, our Monopogen is able to actually reasonably classify those those cells according to their tumor or healthy origin, but other tools had some challenge in doing this, including schematic. So this is actually an independent validation of this as well. I mean, I think schematic is likely useful when you actually know those mutations are specific to particular cell cluster. When you have that information, I think schematic would be useful. But when you don't have that assumption, because mutations can present in multiple different cell type or cell cluster, right? Particularly in cancer or maybe in Alzheimer's disease, for example, then I think our tool would have a tremendous small advantage in those settings. Okay. Lastly, I'd just like to show you one example of Monopogen not only can impute data, but can also make new discovery, sometimes. For example, this is a tumor sample from uterine serous carcinoma sample. This is a RNA UMAP. As you can see, there's 2 cluster green of unciliated epithelial cells here. And we perform Monopogen analysis. This is 3' RNA sequencing. And we detected 24 putative somatic SMVs. And one particular SMV, very interesting, is enriched in one of the epithelial groups, unciliated epithelial group, but not the other. So this is actually very striking difference between those 2 groups. And this mutation actually turns out to be a stop-loss mutation in this particular gene, NME2. They were annotated as a potentially driver mutations, and they have been found in cosmic database in many other cancer types as well. Okay, and now you actually saw the SMVs co-segregated with a particular transcriptomic states. And if you look at this is again showing the transcriptomic or expression level of this particular gene harbors mutation actually is higher when they have the mutation than when they don't have the mutation. So there's a kind of a double hit thing here, indicating the mutations potentially associated with gain of function or maybe increasing of the gene expression. Indeed, if you actually do a differential gene expression analysis contrasting those 2 groups, you can say the first group, the red cell here, group 1 without actually, the color may be a little, this group without mutation actually is mostly has a signature of RNA splicing and histone methylation epigenetic modification. But the one with the mutation actually has much more tumorigenic feature like uncle OXPHOS and MYC amplification, So basically this indicating this is likely to have a higher tumorigenic potential. And this one is potentially still relatively, normal. So there's a kind of 2 pathway of evolution. And this mutation is potentially a driver mutation associated with the evolution of this particular cluster of cells. Okay.
So, Dr. Chen,
Yes.
so
Yes.
I saw there are some blue cells in the red cluster and red cell in the blue cluster. I mean, in the leftmost figure. So do you think it's just some, coding artifacts in some single cells, or do you feel it's a real biological phenomenon?
That's a hard question. Yeah, we don't know yet, but that's a great catch. Yeah, it could be either, I think most of those are likely lack of coverage or something like that, I would say. But it could be biology as well. We haven't followed up at that level. I think it would be very interesting if they're actually real biology. Yes, there's another Ash.
Yeah, hi. So I do have a question on the UMAP here. So like there are a few of the cells, those are gray and those are not selected for the group like 1 and 2. Can you like tell us why that is there?
Oh, actually, gray are the one who has a coverage lower than we're able to analyze, I think. Those are right, Jane. Those are kind of a no reads. Yeah, the gray are actually zero reads in the cell, or
Okay.
zero detectable reads in the cell. So those are no call.
Okay. Thank you.
Yeah, so we're trying to delineate those with data, without data. Okay, so this is actually interesting example showing that just from single cell RNA sequencing data itself, you actually have a very opportunity to actually perform multi-omics analysis to identify some putative, very promising, potentially driver mutations in those contexts. Okay, just like to quickly summarize what I just talked now. So we developed this tool, now published Nature Biotech. The code freely available, and I will actively maintain the code. Actually, the first author, Jing, who has done a lot of job to make the code user-friendly and also efficient. We're still optimizing some of those. So if you use the tool, you have some issue, feel free to reach out to us, or we'll try to solve the problem, technical problem for you. And I think the main achievement here is we're able to reliably call germline SNVs, and sensitivity is typically greater than 20%, and specificity is very high, roughly 95%. Mostly cover non-coding region SNPs, and they are sufficient for genetic ancestry mapping, even on the kind of a difficult, very ancestral similar populations. We're able to delineate them, able to enable QTL analysis at cell type or even cell level, if in some instance. We're able to cause somatic mutations to resolve clonal architecture, and in some instance can discover potential driver mutations. So we're actually still actively developing this tool. So we're trying to, as I showing you one example, we're expanding this to indel and other type of variants. And we have examples where this can work on spatial sequencing data as well, but it's very preliminary. I'm not going to show the data today. We're trying to see if we are dealing with copy number alteration overlapping with SNV a little bit better, because right now, like InfraSNV, copycat, those are kind of separate programs. We're trying to merge our program with those and see if we can jointly characterize those and also perform phylogenetic construction. So those are the kind of thoughts we had in mind, and trying to, potentially even working with you guys if you're interested in collaborating to actually further derive those capabilities. So with that being said, I just like to acknowledge this work is pretty much driven by Dr. Jinzhuang Dou, who's a data scientist in the lab. And also this presentation were enabled by Ziyi Wang, who's a graduate student in the lab. And a lot of our collaboration support from our collaborator labs in the H10 and Chen initiative, and also supported from NCI separate and also MD Anderson one-shots. So with that being said, I'm happy to answer any additional questions you may have.
All right. So, well, thank you so much for the great talk. It's very interesting tool and was a story. Anyone who has a question, please type that in the Q&A chat or just raise your hand. I think August and I will try to unmute you. Okay, we have one question from Chen Yi.
Hi, Dr. Chen. I think this is great talk and fabulous software tour. I'm wondering, because I think this is using the single cell data, and those are mostly limited to 5' and 3' end. So would the variants like those would be limited to the like the near the end, right? They won't cover if any variants of interest would be in the middle of the
that I think that is still very much true. So I think they still have relatively limited coverage in the exonic region, admittedly. I mean, one example I shown here, I think your understanding observation is correct, online with us. As I can show here, just come back to comparison of what we can detect from whole exon sequencing with single cell sequencing, we're able to only detect 40% of the coding SNP that can be detected from the whole exon sequencing. So roughly 40% of exon is well covered for the SNP SNV coding purpose, but 60% is still not being fully covered. This is of course the 10x 5' 3' chemistry, not the what's the other technology, the whole transcriptomic.
Yeah. Right.
Yeah.
Yeah, I just wonder, yeah, if you are also considering like applying this to like those long read single cell technology.
Yeah, long reads is definitely one thing we're applying to, or started to trying to develop this into. And even for the actually SmartSeq, yeah, I was trying to refer to SmartSeq. For the SmartSeq chemistry, which has less number of cells but broader transcriptomic coverage, we also applied our tool to. I think in those chemistry we can do a lot better in the exonic region, although the number of cells are relatively limited. So therefore, there's limited impact in applying or in study those type of data. But yes, but thanks for the question.
Thank you.
Okay, I think I also have one more question. If no one have question in the chat, looks like not. So the question is for your future plan. So the variance calling using the spatial transcriptomics data. I was just wondering, like we have different technologies here, like 10x VZIM, Murphy's, Cosmic, So if we are going to use your tool to do the variant calling in the future, which technology do you recommend to use?
Yeah, so this is a backup slides. Basically, we tried on the SlideSeq DNA and RNA-seq data. This was developed by Chen, as you guys know, at MIT. So we're able to detect those variants that is help us spatially co-segregate, delineate some of the clones, spatial clones. So this, I think, is the most relevant technology at this point, because many spatial technology are not sequencing based, they are imaging based, so they're probe based. So those ones don't have the sequence coverage to call mutations. So therefore, we have to, I really hope this SlideSeq and SlideTech, this technology can become more widely available. So that's where I think our tools can be will be applicable to. So my view.
Great.
Yeah, I have another question. So did you try to apply your tool to some non-cancer samples, like the brain samples? Because I guess in the normal brain samples, the somatic mutation rate will be much lower, and also the clone, the mutant clone will also, they have a relatively small clone, so I guess the refraction will be also low to detect.
Yeah. So that's a great question. We have some ongoing collaboration in that space. I can talk to you offline, but
Sure.
we have also been applied this to what we call pre-cancer tissue, pre-cancer lesion. This is actually a project from pre-cancer. Those are, as you know, much like hyperdysplasia, those kind of sample. They are not yet aggressive cancer or adenocarcinoma sometimes. So we're able to saw a mutational burden or mutational rate difference across different stages of those tissues. So at least in terms of mutational burden characterization, we're able to saw some interesting indication of the somatic mutation detection seems to be successful. But I can't, I've not validated those yet, so I cannot, I want to refrain myself from over-promising. So, but
you can
that's
also
a good
check
question.
you can also check the spectrum, the mutational spectrum, to see whether they look similar to the whole exome or whole genome identified mutation. So that's another way to benchmark the performance. Yeah, because we try some single cell RNA brain single cell RNA-seq data using the SCOmatic, but it doesn't work.
Oh, sorry,
all the mutational spectrum doesn't look correct, and also the mutation burden is also much higher than expectations. So we are eager to see if there are any other tool like your tool could be doable, work on those samples.
That's a great
Yeah, maybe we
suggestion.
can try offline.
Yeah, if we have some question, we can maybe connect to you as well. We haven't done much of that level of QC, but sounds like a great idea to do.
Great. I think that we don't have more questions right now. Maybe we can stop here. Thank you so much for the great talk. I think that's really, we actually know a lot from your presentation. I hope that, the audience will try to use your tool in the future and get back to you if we have any question.